everyone, I am Dr. Sonal Chain from Department of Anthropology, University of Delhi. In this series of forensic science lectures, today I am going to talk about various immunochemical techniques. In forensic science, whenever we found a biological sample at the crime scene, there are various ways we can investigate the sample to know about what type of sample is that. We go for various analysis. For example, we go for certain preliminary analysis and sometimes we go for certain confirmatory techniques. When we go for certain preliminary analysis, there are certain techniques, immunochemical techniques we use for, and for confirmatory techniques also we can use certain immunochemical techniques. The most widely used biological sample found at the crime scene which can be analyzed using immunochemical techniques is blood. For example, if we have found blood at a crime scene, firstly we should know whether the substance which is present or which is being found at the crime scene is any other red as a substance or is it blood. So, for knowing that, we go for certain preliminary analysis, for example certain microscopic analysis, and we can see under the slides by using certain slides test, microscopic test, Takayama-Tickman test to confirm that the substance, the red substance which is found at the crime scene is blood. But the second main question is whether the blood which is found at the crime scene is human's blood or belongs to another species. So, for that particular question we can use immunochemical techniques. That is one of the example where these immunochemical techniques can be used. There are a lot of immunochemical techniques and as per the requirement they are used at different stages. So, to start with, immunochemical techniques are based on a reaction of antigen with antibody, or more exactly on a reaction of an antigenic determinants with the binding site of the antibody. The antibody used are produced by various ways. Antigen-antibody reactions, antigens and antibodies combine with each other specifically and in an observable manner. In the body, they form the basis of antibody-mediated immunity in infectious diseases or hypersensitivity and autoimmune diseases. That is, we say that if there is an infection which is coming from some foreign substance in our body, our antibodies are there which will fight for that. That happens in the antibody and the antigen we will consider which is coming as a foreign substance and our immune system will fight with that. But in the laboratory, these antigen-antibody reactions, they help in various ways in the diagnosis of infection, in epidemiological surveys, in the identification of infectious agents, enzymes, and also for species identification of various blood or other biological fluids. So, antigen-antibody reactions in vitro are known as serological reactions. They fall under forensic serology. There are three stages of antigen-antibody reactions. For antigen, we will be using Ag as the small form and Ab for the antibodies. So, at the primary stage when we talk about, the initial interaction between antigen and antibody is invisible. Whenever antigen and antibody they combine with each other, we see nothing. Then rapid occurs at low temperature and obeys the general laws of physical chemistry and thermodynamics. This reaction is reversible. Antigen and antibody is bound to each other by weak van der Waals forces, ionic bonds and hydrogen bonding. So, whenever any primary stage reaction happens, there is nothing, no such antigen antibody reaction we will see, that the reaction will be invisible. Going to the secondary stage, there are certain demonstrable events in the secondary stage. For example, we can see the precipitation, agglutination, lysis of cells, killing of live antigens, neutralization of toxins, complement fixation, immobilization of motile organisms and enhancement of phagocytosis. So, In the secondary stage, these type of reactions occur. Precipitin is when antibody participate in precipitation, then we can see a coagulative white color kind of formation. So, we can say a precipitin formation is obtained. Whereas agglutinin is known as when antibody participate in agglutination reaction. Similarly, we can see a kind of agglutination which is also a kind of white foamy thing. Precipitinogen is the antigen participating in precipitation, then we call it as precipitinogen, and agglutinogen when antigen participated in agglutination. So, depending upon whether the antibody is participating in the precipitation reaction or the antigen is participating in the precipitation or agglutination reaction, we can name the product formed. Antibody participation, we name it precipitin or agglutinin, and if antigen is participating, we name it as precipitinogen or agglutinogen. These are the examples of secondary stage reactions of antigen and antibody. Coming to the tertiary stage, it includes neutralization or destruction of injurious agents or tissue damage. It also includes the humoral immunity against infectious diseases as well as clinical energy and other immunological diseases. So, there are general features of antigen and antibody reactions. The reaction is specific, entire molecules react and not the fragments. There is no degeneration of the antigen or antibody during the reaction. The combination occurs at the surface. So, surface antigens are immunologically relevant. The combination is firm but reversible. The firmness is influenced by the affinity and avidity of the reaction. Now, as we say, the reactions are specific. So, that means if that particular antibody is formed by or is against that particular antigen, then only the reaction will happen. For example, if we consider that the blood which we have found at the crime scene is of any animal species. So, supposedly that blood, if we treat as an antibody and if we put antigens of different animals, then only the antigens of those particular species of animals will react with the blood to whom that particular blood belongs to. So, the reaction is very specific and it is a confirmatory test confirming the species reaction. Now, as we spoke about the firmness of the reaction, which is influenced by the affinity and the avidity of the reaction, we must know what is affinity and what is avidity. So, affinity refers to the intensity of attraction between the antigen and antibody molecules. It is the function of closeness of fit between the epitope and antigen binding regions of its antibody. Whereas avidity, as we say, it is the strength of the bond after the formation of antigen antibody complexes is there. So, avidity is determined when the reaction is completed. When we say that antigen antibody complex is formed, the strength of that bond determines or says about the avidity, whereas the affinity says about how much is the attraction between that particular antigen and that particular antibody. So, for making a precipitate or for making an agglutination, antigen and antibody, they both participate in the reaction. In various reactions, there are various ways of putting the antigen and antibody. They are combined in various proportion. Sometimes the reaction can happen without the use of any external force, but in certain cases we require external force for such reactions, where an external current is being passed to make the reaction possible. How do we measure antigen and antibody? Measurement may be in terms of mass or more commonly as units of titer. The antibody titer of a serum is the highest dilution of the serum which shows an observable reactions with the antigen in a particular test. So, we have to consider whenever we are going for an antigen or an antibody reaction, how much antibody is required for that reaction and how much antigen is required for that reaction, for an observable reaction. If we are taking very less amount of antigen and very less amount of antibody, then the reaction results will not be observable to our naked eyes. So, we have to define a quantity of antigen and antibody for an observable reaction in a particular test. There are two important parameters in serological test. One is sensitivity and one is specificity. This is just not about the serological test. Any test we perform in any science, the sensitivity and specificity are the two most important parameters. Sensitivity is the ability of the test to detect even very minute quantities of antigen or antibody is present. When the test is highly sensitive, the false negative results will be absent or they will be very, very low. Specificity, the ability of the test to detect reactions between homologous antigens and antibodies only and with no other. In highly specific test, false positive reactions are absent or minimal or lowest. So, for a technique, if we say that this is the best technique or this is the best test for particular analysis, for identification of a particular substance, then that test should be highly sensitive and highly specific. We say for highly sensitive, the test, even if the minutest quantity is present, the test should identify that particular antigen or antibody present. It is in case of any test. Supposedly, if we are using any alcohol test and we are going for gas chromatography, then the technique should be highly sensitive to identify those alcohol particles. Specific, that the technique should not give result with any other substance, only with the homologous substances. So, highly specific and highly sensitivity are the two important parameters in any test. There are various types of antigen and antibody reactions: precipitation reaction, agglutination reaction, neutralization reaction and complement fixation test. Immobilization test, opsonization, immunofluorescence, radioimmunoassay, enzyme immunoassay. So, we will be discussing one by one all these various tests and how they are used for the analysis of various biological substances or various chemical substances which are found at the crime scene. So, first starting with the precipitation reaction. The principle of this reaction is whenever a soluble antigen combines with its antibody in the presence of an electrolyte at a suitable temperature and pH, the antigen antibody complex forms an insoluble precipitate. When instead of sedimenting, the precipitate remains suspended as flocculus, it is known as flocculation reaction. Precipitation can take place in liquid media or in gels such as agar, agarose or polyacrylamide gel. So, for a precipitation reaction to happen, the antigen and antibodies, they should combine at a specific temperature and pH, and we require a liquid media mostly in cases we use agar or agarose gel or any other polyacrylamide gel for the precipitation reactions. So, whenever we are talking about the precipitation reaction, there is one phenomena known as zone phenomena. The amount of precipitate formed is greatly influenced by the relative proportions of antigens and antibodies. For example, if the amount of antigen and antibody is very high and they are homologous in nature, then a strong precipitate will form. If the antigen and antibodies are not homologous, then there will be no precipitate, and sometimes if the amount of antigen and antibody is very less, then a very light precipitate will form. That's why whenever we were talking about the measure of antigen and antibody reactions, we should take appropriate amount of antigen and antibody so that a visualized reaction should be available or observable reaction should be visible to the naked eye. If increasing quantities of antigens are added to the same amount of antiserum in different tubes, precipitation is found to occur most rapidly and abundantly in the middle tubes. For example, in the preceding tubes, if antibody is excess, that is known as the reaction will occur at pro zone. Wherever the amount is excess, the reaction will occur there. It is known as pro zone. In the middle tubes, antigen and antibody in equivalent proportion, then we call it zone of equivalence. For example, we have a ring tube test. So we have put antibodies at the base, antigen at the top, and their quantity is similar. So the antigen and antibody, they will form a ring in between. That is known as zone of equivalence. In the later tubes, if the antigen is put excess, then the reaction will perform at the excess site, the precipitation will form at the excess site, and that is known as the post zone. So, the reactions can occur in pro zone or in the zone of equivalence or the post zone, depending upon how much amount of antigen and antibodies we are using. So, we can see in the image that with the increasing antigen and with the increasing antibody, if we go towards the right, the antigen will be increasing, and if we go towards the up, the antibodies will be increasing. So, the antibody anti-reactions, this is the zone of antibody excess, the first one, which we also call as the pre-zone, where the zone of equivalence, where the equal amount of antigen and antibodies are there. So, the reaction will be happening there, but if the amount of antigen is more and antibodies are less, that is known as zone of antigen axis, that is known as prozone, and the reaction will be happening in such cases at the prozone. Who developed the mechanism of precipitation? Marek in the year 1934 proposed the lattice hypothesis, also known as the mechanism of precipitation. The multivalent antigens combines with bivalent antibodies in varying proportions depending on the antigen-antibody ratio on the reacting mixtures. Precipitation results when a large lattice is formed consisting of alternating antigen and antibodies. So, Marek's also show the equivalence zone according to Marek's hypothesis, where antibody is there, the excess zone, then we have the pre-zone, zone of equivalence where the amount of antigen and antibody is similar, and the antigen excess zone where then remaining antigen will be left out and antibodies will react with the antigens which are present there. So, the red one is the pre-zone, purple one is the zone of equivalence, and the blue one is the zone of the zone where antigen are excess in nature. Now, we talk about the application of precipitation reactions. They can be carried out as either a quantitative test or qualitative test. So, whenever we are talking about antigen and antibody reactions, we need to find out two things. The first thing is whether the reaction is happening between the antigen and antibody or not. Like we can say the antigens are homologous or antibodies are homologous to each other or not. So, that is a qualitative test telling us that the particular antigen and antibodies are homologous to each other. But in certain cases, we can also determine the amount of antigen or antibodies present. In such cases, we have to send the labeled antigen or antibodies. Depending upon how much antigen or antibodies are used up and how much are left out, we can quantitatively tell the amount of antigen and antibody present in such reactions. So, the precipitation reactions can be either qualitative in nature and quantitative in nature, depending upon what kind of results we are looking for. They are sensitive for the detection of antigens. If antibodies are put in a precipitation reactions, we can easily identify and look for the antigens. The types of precipitation reactions are ring test, slide test, tube test, immunodiffusion, electroimmunodiffusion. So, the first one is ring test. It consists of layering antigen solution over a column of antisera in a narrow tube. For example, we have a test tube in our hand. In that test tube, we can put a layer of antisera, and over that we can put a layer of antigen, and then precipitation reaction can happen in that tube only. For example, thermoprecipitin test, grouping of streptolocci by Lenser field technique, etc. We can see in the image that ring test how it is happening. In a normal test tube, we have put some antibodies which we are looking for, that they might be homologous to the antigen which we have to identify, and on the top of that we put the antigens which have to be identified. In the between, the precipitate is being formed. If the precipitation has happened, a precipitate is being formed, then it means that the antigen and antibody are complementary or homologous to each other. If there is no precipitation formation in between, then we can easily say that antigen and antibodies are not homologous. For example, if we have found any specific blood at a crime scene and we are suspecting that it's a case of wildlife poaching, and we are suspecting that the antibodies which are present or the blood which is present is of any tiger, lion, and we are putting the antibodies of that particular animal. If the precipitation reaction is forming, then we can confirm that in the wildlife poaching case, if that particular animal is tiger or lion. So, by using these simple tests, ring test, precipitation test, we can easily go for the preliminary as well as confirmatory analysis which are required and can be used in forensic cases at simple steps. So, to conclude, I will say that precipitation reactions are very important and can be used as presumptive and confirmative tests in various forensic serological cases. Thank you.